So today we're going to be talking about developmental networks and how they look when there are mutations affecting them. So in spikes in dragon cells, we have this pathway that is S promotes P blocks I blocks K, which promotes E and ends up promoting spikes. So the wild type will end up having positive spikes because P is going to block inhibitor I, which allows K and E to exhibit spikes. But if P is not present, as in there's a P minus, then inhibitor I is available to block activator K, which promotes E and eventually spikes. So pretty much no P, so I is present to block K, and so no spikes are formed. When I is not present, then spikes are formed because I is not there to block K to E to spikes. When I and E are not present, the one that kind of matters is E, and if E is not there to promote spikes, then spikes are not going to be formed. And if S and K are not present, then there are not going to be spikes. And again, we're going to look at the protein that's further upstream, which is going to be K, or sorry, further downstream, which is going to be K. And since K is not present, it cannot promote E, which promotes spikes. And so therefore spikes will not be formed in that cell. And so for a type of regulation that can occur to make this type of chain happen with no spikes being formed due to a lack of presence of the proteins, we can see that in histone modification, which regulates genotype. And so pretty much when HDAC is present, the histone will be deacetylated and the histone will be tightened. This will make it harder for the DNA to be read by DNA-P or DNA polymerase, and so it cannot be transcribed and expression is muted. If the histones are deacetylated, then the DNA is wrapped more tightly around the histone complex, and so DNA polymerase cannot go ahead and read around the DNA, and so the proteins that are being used to transcribe or to make the spikes are not going to be able to be transcribed, and so the expression of spikes will be muted. And here are my references. So thank you.